this the ROM Toronto Instagram account? Hi everyone, nice to see you all. This is the ROMKids Show. If you're here, that means you want to learn about DNA today. Today on the show, technician at the ROM, DNA expert, Oliver Haddrath is here to talk to us about DNA, what it is, and why it's the building blocks of life. Then we're going to do a science experiment where we are going to extract the DNA from a strawberry. And if you're asking today, Kieran, what's the theme with your socks? Because you're always so on theme with your socks. Batman. Well, Batman based on bats. Bats love fruit, and we are featuring a fruit today, strawberries. So that's the connection there. Hi everyone, so nice to have you here. If you're in a classroom, hello, hello. If you are in your home, hello, hello. Again, over the next sort of 30 minutes, we're going to do a science experiment, talk about DNA with our special guest, and just have a conversation. So if that's what you're interested in, hang out with us. Remember, all shows go up on YouTube. You can see last week's episode with David Evans where we talked about dinosaurs and we made some silhouette dinosaur art up on YouTube right now. And of course, we also did our episode at the start of season 3 with educator Julie Tomei, where we talked all about the moon and we made our moon piece. So all that's up on the ROM at Home website. I think I'm ready to go. It is the theme song portion of the event. Let's get started. Welcome to the ROMKids Show with me. We'll do some crafts and tell some stories. Let's talk about science, art, and history. Welcome to the ROMKids Show, starring you and me. Nice, that's the theme song portion of the event. Enjoyed that, that was a lot of fun. Let's move on over to the art table and get set up for the day. I'm feeling particularly proud about that performance of the ROMKids Show theme. That felt good. All right, are we all here? Are we all ready? What do you need to collect right now so that you can do this science experiment with us? So today we're doing the DNA extraction experiment. We will be extracting our DNA from a strawberry, okay? Now, it's the middle of winter in Toronto, which is where we're doing the show from. So this is a frozen strawberry. This frozen strawberry is going to come in very important in our narrative later in the show, because I've just learned some hot tea about why using a frozen strawberry is better for this project. So what do you need? You need a strawberry, okay? Next, you need, and I'm just making sure I'm doing this right. I wrote it all down. You need 90 milliliters of water, okay? So I measured it out right beforehand, 90 milliliters of water to make our solution. You need 1 teaspoon of rubbing alcohol, and it needs to be like super cold. So put it in your freezer right now. Ours has been in our freezer overnight, so do that. Make sure your rubbing alcohol is in the freezer. We're gonna need 1 teaspoon of that. We're gonna need 2 teaspoons of dish soap. We're gonna need 1/4 teaspoon of salt. And I pre-poured this because I have a really bad habit on this show with salt and pouring it live and it getting everywhere. You're going to need a Ziploc bag. I got these giant ones. This is where we're gonna smush everything up to make our strawberry solution. You're gonna need a cup to mix everything in, kind of key. You're going to need then a sort of sift, I'm calling it a sift, a strainer, to put all your solution through in a bit. And then you're gonna need your little final cup to pull the DNA from, and then tweezers, or we're using a bamboo skewer, just to be able to lift the DNA out. Now, I've done this experiment many times in my life, and I gotta say, I have a lot of questions about it. So today on the show, to answer our questions, we have our good friend, Oliver Haddrath with us. Welcome to the show.
Hey, thank you, Karen. Happy to be here.
Oh, this is so exciting. I've always had a lot of questions about this because, we know DNA is like, we can't really see it with our eyes. So being able to do an experiment where we can is fascinating. So while I got you here, we're going to do the very first step together, and then we're going to get into our questions. And so friends at home, if you have questions about DNA, throw them into the group chat right below, and we'll ask them with our friend Oliver in a bit. So the first thing is make sure you have adult supervision. Grab an adult to do this adventure with, whether it's your teacher or someone in your home, a trusted adult, to make sure your experiment is done safely. Remember, we are using rubbing alcohol later in this process, so safety is important. And our next step is we need to make our DNA extraction mix, all right, which is just really cool words to use very basic things. So what you're gonna do is you're gonna pour your water, which I have my water right here, into your mixing cup, which is right here. I'm just gonna pour that water right in. Love that for me. Then what you're going to do is you're gonna add your salt. So you're doing 1/4 teaspoon of salt into your 90 milliliters of water. Just gonna drop that right like that, a little splash, that was fun. And then you're gonna take 2 teaspoons of dish soap, okay? And so I'm just gonna pump right in. Now, Oliver, why am I using, we're doing a DNA experiment, why am I using dish soap, water, and salt?
Well, the whole idea of using the dish soap is because soap actually can help break up cell membranes. We're all made up of trillions of cells, and they're all contained in a membrane. And the soap breaks open the membrane and releases kind of the guts of the cells. Now, the DNA is a charged molecule, and it's dissolved in the water. Salt is also a charged molecule when it's dissolved in water. So you add the salt, it'll help neutralize the charge on the DNA. And what that means is it'll be easier to bring the DNA out of the water, out of the solution.
That's fascinating.
Because ordinarily it's dissolved and you don't see it.
Huh. Okay, cool. I've always wanted to know the answer for that. Okay, so while you're grabbing all your materials and doing that first step, making your extraction mixture, we need to talk to our friend Oliver about DNA. And my first question is an easy one, but maybe actually really hard. What is DNA?
DNA is a molecule you find in every cell of your body, well, with the exception of red blood cells. And it's kind of like the computer code. Your cell phone or your computer, it runs because you have a program or an app that gives it instructions on what to do. And then the phone or computer does whatever you tell it to do, or the program does. The DNA is the molecule in the cell that tells the rest of the cell what to do. Encoded in it, in these bases, are the instructions on how to make the cell work and run and do everything it needs to stay alive.
Okay, so my follow-up question to that is, how does, I'm thinking about my tongue, and there's DNA that needed to turn that into a tongue, okay? So thinking about my tongue right now, how does the DNA know that it's going to be a tongue?
That gets a little complicated because every cell of your body that has DNA has a complete program, a complete blueprint for making every part of your body. But in different parts of your body, like your tongue or your eye, only certain parts of the DNA are being read. So it knows it's the tongue, so it's going to express the genes or the bits of the code that are involved in making the tongue do what it has to do.
That's fascinating. So each DNA can do anything in the body, but based on what it's interacting with is how it knows to be what it's going to be. Wow, I'm already learning so much. Okay, I do have another question about DNA and like size, okay? So like it's a molecule, so that means it's really, really small. Like I can't see it with my eye. Is DNA different in size for, say, like a giant tree? Is the DNA in a giant tree bigger than the DNA in like a tiny hamster?
Well, you see, this is where it gets really strange. Small things tend to have smaller stretches of DNA. A human's DNA, total DNA, they call it the genome. Its total DNA is about 3 billion base pairs long. So that's a lot. Now, if you look at something like a bird, it's only 1 billion base pairs long. If you look at something like a fly, like a fruit fly, it's only something like 300 million. If you look at a bacteria, it could be just a few million. If you know we have this pandemic going on with COVID-19, it doesn't use DNA, it uses a related molecule called RNA. And the entire computer code that's in the RNA is only 32,000 bases long. So it's very, very small. So you start to think, the bigger, the more complex, the larger the genome, the more code you need. But nature is always interesting because it always throws us for a loop. The human genome is 3 billion base pairs long. That's how long an onion's genome is.
100,000.
15 billion.
What?
So an onion has basically 5 times as much DNA as we do. And a salamander can have 120 billion base pairs. So some organisms have a lot more DNA than others. But what they think is that not all that DNA actually does anything. Some of it might just be kind of filler or spacer. For instance, a mammal has about the same number of genes, and these are bits of the DNA that do things, as a bird. But a bird only has 1/3 of the amount of DNA that we have. So it's still pretty complicated, and people are still trying to figure it out. The general rule, the more complicated the organism or the animal or plant, the more DNA it needs to run. But it's not necessarily size, because if you take a blue whale, which is the biggest animal that's ever lived, its DNA, its genome, is smaller than ours.
Huh.
So we actually have more DNA to make us than it takes to make a blue whale.
And the base pair of an onion is 15 billion. And sorry, how many for a human?
3.1 billion.
Wow.
So there's a lot more DNA in an onion.
That's so interesting. That's so interesting how, some what we consider to be like complex forms, such as like you or I and our friends at home and at school, then compare it to an onion. I would have thought I would have more.
Yeah, I know you'd think that, but that's where nature gets kind of interesting. And we're still trying to figure it all out.
I'm already learning so much. Okay, we're gonna do the next step of our experiment here. So we've made our extraction mixture. So what do we do next is we're going to take our Ziploc bag, all right? I'm then going to take my strawberry, which again was frozen, so it's a little like melty. I'm going to put my strawberry right in there and hope I don't spill any, because that would be bad, because we have a white carpet right underneath, and everyone, I nailed it. I didn't spill any. Then we're going to take our extraction mixture and we're going to pour it into the bag. Woo! Okay, get all of that in there. Again, these household materials are going to allow you to see something that is generally, it's a molecule. You can't see it with your naked eye, and we're going to do that today. So then you're going to close up your bag and try and get out as much air as possible. Because if it has air in it, then you might explode it. It might pop, okay? So important for us to do that. And then you're going to seal it as tightly as you can, because here's the fun part, all right? The fun part is we're going to spend like the next 4 to 5 minutes just squishing this bag, okay? Just squishing the bag. Once you get everything in, you're literally just going to start squishing away at your strawberry, okay? See how I've started to like mix it up? You don't want it to look anything like a strawberry anymore. In fact, you want it to look almost like a strawberry jam, okay? So squish, 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 and let's move to our next question. We know we can't see DNA because it's so small. So when you study DNA, how in like your lab, and I got a picture of you in your lab right here doing cool stuff. Look at this cool dude in a lab. And Oliver has one of the cooler labs at the ROM. That's the one I got from you right there.
Okay.
How do you study the DNA?
Well, the way we actually study the DNA is first we have to extract the DNA, just like you're doing with the strawberry right now. And we can do it from blood samples. If you catch a migrating bird or something and take a tiny blood sample and band the bird and release it, you can actually extract the DNA from the blood. And then we have in the museum machines called, and it's a complicated name, but DNA thermal cyclers. And what they do is, if you give it a tiny bit of DNA, it'll make copies of it, almost like a photocopier. It'll make millions and millions of copies, so you have so much, you can actually study it. You can do things like sequence it. And what sequence is where you read all those base pairs that make up the DNA. You know that the DNA is like a long code? You're actually reading the code. And then you can be able to study it and compare that code between different animals to find out how those animals are related to each other.
Huh. Okay, I have another question, shifting back to like the science experiment that we have right here. What is happening in this bag right now with our salt, with our soap, our water, and our strawberry?
So to be able to get the DNA, because the DNA is really well packaged, it's inside something called a nucleus in the cell. And the nucleus, of course, is inside the cell. So it has another membrane, the cell membrane, going around it. So what you need to do to get at the DNA is you have to mush it up, which breaks it apart. And then the soap comes in and starts to kind of melt away the cell membranes, releasing all the contents of the cells. And the salt will actually be attracted to the DNA because they're both charged, and will help kind of neutralize the charge on the DNA, which will help you later on when you actually want to bring it out of solution. Because right now the DNA is dissolved in the fluids in the cell. Kind of like when you add salt to a glass of water and you see it disappear. What we're going to be doing with the DNA, or you're going to be doing, is the reverse, where you're actually taking it out of solution. So you'll be able to see it. So the more you squish up the strawberry, the more you're actually breaking open cells. So the better you do it, the more cells that break open, and ultimately the more DNA you'll be able to recover. And so you'll see more.
So very quickly, yes, we want this to be a liquid, okay? Because we're squishing and breaking up all the cells so that the DNA can come out. So you can see mine right here. It looks nothing like a strawberry anymore, right? It looks like some jam. It looks like some very liquidy water. Sorry, right there. Yeah, so it should be a liquid, okay? So keep smushing it all up. And like Oliver said, the more it's squished, the more DNA will be released because we're trying to break open the cells. And before we continue, Remy, how's it going? Hope you're doing well. Okay, and if you have more questions, please feel free to drop them into the chat, okay? What I want to do now, okay, because I think we have some big questions that we need to ask in a second, is we are going to, now that we've all squished this all up, is we're going to take it out, all right? And this is another time where Kieran should have thought a little bit more carefully about the fact that there's a white carpet underneath of me. So we're going to try and do this really, really carefully. And we don't have like a better strainer. We just have like this pasta size one. But now that I've squished it all up and you can see like it's just a liquid, is we're now going to take it out and we're going to pour it back into our little jar, okay? And we're going to try and do this very, very carefully. And then we're going to ask some questions that are really on all our mind, such as, can dinosaurs really be brought back through DNA? And so I'm just going to use all of my concentration here to focus on doing this part because I don't want to get in trouble later. Oh, slam dunk. All right in there. I love that. Then you can very carefully push that aside. Okay. And then the next thing you're going to do with a spoon is you're going to tamp on top of your strawberry mixture, okay? And that's going to further break it up and push more of the DNA down into your jar, okay? So I'm just going to be doing this for a little bit, and we're going to go back to talking to Oliver. So before the show, you know, in our pre-interview, we were talking about how DNA, like you can learn a lot about a person by looking at their DNA. But what was really interesting is you were telling me that we can learn not just about one person, but also like populations or groups from DNA. How does that work? Because I thought it was just inside of me only.
Well, because your DNA comes half from your mom and half from your dad. And if you think about it, their DNA came from half of their mom and half of their dad. So in fact, not only are you half your mom and half your dad, you're actually 1/4 of each of your grandparents. But then if you think about it, you're 1/8 of your great grandparents. So in fact, our DNA comes from all our ancestors and it makes us up. So if we actually sequence the DNA, you can compare a lot of the samples across or the sequences and start to learn about how large a population you actually came from. We're doing a project in the museum right now where we're sequencing the blue whale genome. And one of the things we'll be able to answer, like all the DNA that makes up a blue whale, one of the questions we can answer is blue whales were almost hunted to extinction. Now they're starting to make a comeback. The numbers are starting to recover. And some people are wondering, well, how does that compare to how many blue whales there were before hunting? Well, nobody really knows how many blue whales there were before hunting. But if you look at the DNA of modern day blue whales, you can actually look backwards in time and look at how big the population of blue whales were before hunting started.
That's so interesting. So, my DNA is made up of my mom and my dad, and that's maybe what I would think about. I think of me, then I think about my parents, then there's grandma and grandpa, Nono and Nona. All of those things are in there. That's so interesting how you can do that. Okay, so a popular question that we had David Evans on the show last week talking about dinosaurs. You can see it up on YouTube. Also, hi Mylan, how's it going? Is can we bring dinosaurs back? We've seen it in who knows how many movies now as like a thing that can happen. We have a DNA expert here, Oliver. Tell us once and for all, can we bring dinosaurs back from DNA?
At the moment, no. I mean, to be able to use DNA to bring something back, you have to be able to get its DNA. And way back in the late 1980s and early 1990s, when Jurassic Park books and movies were made, people thought they might be able to recover DNA that far back in time, because DNA lasts a long, long time if it's properly preserved. In fact, the oldest DNA recovered now, I think, is just over 1 million years old. So it's possible to sequence the DNA from mammoths, woolly rhinoceros, saber cats. And there is actually the talk that maybe they could bring back a mammoth one day or some other extinct animals. But until you can recover DNA from a dinosaur fossil or remains, then you don't have the opportunity to bring it back. I don't want to say never, because science always finds a way of making great discoveries. Because 20 years ago, I would have said it's very unlikely that we'll ever see a living mammoth again. Today, I don't say that. I think there's a good chance we'll have something that's close, not exactly, but close to a mammoth sometime in the future.
So, big thing I just learned right there is we've only been able to recover DNA going back 1 million years. And if we can recall, non-avian dinosaurs existed 66 million years ago. That was the last time they were around. So I think friends at home, if you really want to see a dinosaur, go look outside, find a pigeon, find a seagull, find a goose. These are relatives. These are dinosaurs that live today. You might even have some tonight if you have some chicken fingers for dinner, or some nuggets. One of those things. Okay, a really big thing that we like to talk about on this show, though, is that maybe we can bring things back from the past, but shouldn't we do a lot of work to save things that are around today? How can studying DNA of animals today help us take care of them?
Studying DNA is really important for conservation. You can actually, for one thing, if you have a species that's in danger and there's a few remnant populations around, you can actually look at their DNA to find out how different they are. We did a research project on kiwi of New Zealand. Kiwis are small flightless birds, and they're endangered of going extinct. Some predictions were they'd be extinct by the end of the century. So they're doing a lot of conservation work, breeding, captive breeding, to prevent them from going extinct. The good thing about looking at the DNA is when we looked at them, we actually found out that the brown kiwi is not one species, but actually made up of more than 2 species.
Whoa.
So when you're doing captive breeding, you don't want to put two birds that are not the same species and try and get them to have offspring. Keep the species going because you're making things that don't occur in nature. So DNA really can help by reading the sequences. You can work out how these animals or plants or whatever they are related to each other. And that can make you, when you come up with a plan to try and save them, make sure it's really informed that you got your best chance of actually succeeding.
That's awesome. And looking at DNA showed you that these kiwis, that there were more than one. And to your point, yeah, you don't want to mix them up because you want to be able to keep the animal to you know, to what it really is and not create some new ones. Okay, so now I'm keeping up my good job of trying not to make too much of a mess. I'm somewhat succeeding. We're going to go to our final part, okay? So now that you've tamped everything down and you should have your sort of your mixture here, what I'm going to do is I'm going to put it into even smaller of a jar, okay? That'll help focus all my liquid together, and then by doing so, sort of keep all of the DNA closer together too. So I'm just going to pour some of that in. Oh yeah, look at that. It looks so good, and that's why you have an adult there telling you you are not allowed to drink this, okay? Do not drink this, do not consume this in any way. So next step, remember that rubbing alcohol from the top of the show that you put in the freezer to get really, really cold? You're now gonna put a teaspoon and pour it into your mix, all right? So I filled up a teaspoon. Now, Oliver, why did I need the rubbing alcohol to be frozen and then or cold? And then what do you think is going to happen when I pour it into the mix?
Well, what happened is you've now broken up all the cells. You've released all the contents in this solution. You put it through that strainer, so all the stuff, the solid bits, got left behind, and just the contents with the DNA went through. So that red solution is containing material from the strawberry that's still dissolved and the DNA. Now, earlier on, you added the salt, and the salt helped neutralize the charge on the DNA. And what that means is that the salt is not as easily dissolved anymore. It's still dissolved, but it's not as completely dissolved. So when you add the ice cold alcohol on top, it's going to make the DNA come out of solution that suddenly it'll start to appear.
Oh, goodness. Okay, so here we go. We're going to try this out live. I'm going to pour it in, okay? Here we go. Do we feel good? Okay, I've poured it in. And you can see there's some almost like bubbling occurring right on the top. And I'm going to venture a guess that might not be bubbles necessarily, but our DNA getting pulled out. And so here is where you need your toothpick or something to try and pull it all together. Whoa! Look at that. It's like the entire top layer is all pulled up. Do you see that? That's so cool. That is all of our DNA from inside of the strawberry. Nice. That's a good shot. Whoa. So that's our DNA that's been pulled out from our strawberry right there. And so it doesn't look like the double helix that we normally know about a piece of DNA looking like, because this is just a bunch of it pulled together.
Yeah.
Okay, so now I have two more questions about strawberries, and then we're all done for the day. You can see that all right there. Now, I froze the strawberry first, and I found that it released more DNA. Why is that?
Well, you have to work hard, a fresh strawberry, to break it up into all the cells and rupture them. But when you freeze things like plants or strawberries or any other kind of fruits, you'll notice they get soft and gushy because the ice crystals that form inside the cells break them open.
That's so cool.
And so, in fact, freezing them does a lot of the work for you, and actually is better probably at breaking all the cells apart than just grinding it in a Ziploc bag.
Huh. Okay, so that's a
And you
good.
notice what color it is?
It's like translucent and like white almost.
Yeah, and when it dries, it'll go white.
Wow.
And that's the DNA that you're seeing.
That's so cool.
And strawberries work really well for getting their DNA because they're octoploid. And what that means, humans are diploid, which means two. We got one copy of our DNA from mom, one copy of our DNA from dad. Strawberries and some other plants can be a bit weird. They have 8 copies of the DNA. So they have a lot of DNA in their cells. So that's why they're really good ones to use as an example.
That was going to be my other question. You want to use a strawberry. Now, you can do it with like a banana or like a kiwi.
Yeah, or an onion, or most fruits will actually work. Some are just a little harder to break up. Strawberries work really well. And because the DNA is white and the strawberry is red, it's easier to see the DNA pulling apart. With a banana, which is kind of a yellowy white, it's a little harder to see the DNA.
Those are such good tips. I would not have known the banana one. So you can do it with others. It's strawberry because it's an octoploid. It has those
Octoploid.
Octoploid. It has those 8 sets. So there's even more DNA to get pulled out. And then that's it. Oliver.
Yeah. I was going to show you what it looks like when it actually dries. This is about 1/4 gram of DNA. And what's really cool about DNA is it's a really good way of storing information. This 1/4 gram of DNA can store 50 million gigabytes of data.
Whoa.
So that 1/4 gram could store the same amount of information as 1 million iPhones that all have a 50 gigabyte hard drive.
That's awesome.
So a very cool way of storing information.
DNA. Maybe, it's the past, but maybe it's also the future. Folks, that's it. Thank you for joining us from your classrooms, from your homes, from your kitchens. It's another episode of the ROMKids Show. This one all about the strawberry DNA. We are back next week on a very special episode about water, access, and equity. So join us for that. This episode with Oliver will be up on YouTube later in the week. Oliver, thanks for joining us.
Thank you. It was a pleasure to be here.
Friends, have a wonderful week. Stay safe. Wear a mask. We love you. Have a great day. Bye, everyone.
Bye.